In this video, we'll be looking at some previous exam questions, specifically on genetics, and then this one will be based on sex-linked inheritance. So let's look at the information provided and then the questions asked. So after the larva stage, female mayflies usually live less than 5 minutes, while males can live a whopping 2 days. The average female mayfly lays anywhere from 400 to 3,000 eggs. You can often differentiate males and females by their eyes. Males will usually have much larger eyes than females. Males also have claspers beneath their tails that are used to hold the female while they fly and mate. Now, sometimes the information that they give is purely as a little introduction to the question, but sometimes there's some valuable info that they can ask in later questions. So let's get to the first question and see what they want to know. So, in mayflies, the gene for eye color is carried on the X chromosome. Now, immediately there, I can already tell you that this will be a question based on sex-linked inheritance. And the reason I can do that is because of that X chromosome that we see there. So we know that males have XY chromosomes and females have XX. So as soon as we see those chromosomes, we know that it is sex-linked. Now they say the allele for blue eyes with a capital B is dominant. So they did not have to tell us that it is dominant. We could have purely seen that from that allele that is written with a capital B. And they say it's dominant over the allele for yellow eyes, which is shown with a lowercase b. Now they're going to tell us what they want to have crossed. So they are going to cross a yellow-eyed male with a homozygous blue-eyed female. Use a genetic cross to show the F1 generation. So remember that you're going to, since it's sex-linked, you're going to use XY and XX, and then the allele will be written at the top right position of the X chromosomes. Y chromosome, the Y chromosome does not carry an allele, so with males it's just that one X chromosome that can. So let's get started with this genetic cross. I'm assuming that you already know how to do a genetic cross. If you don't, check out the previous videos. So I'm just going to get started straight away. So we always start with P1, so that is the parental generation, so P1, and we will start with phenotype. So what is the P1 phenotype? We are crossing a yellow-eyed male with a homozygous blue-eyed female. Now you do not need to write homozygous, you can just write blue-eyed female, and the reason I say that is because in the genotype, when we write that, they will be able to see that it is homozygous. So genotype, what does the male look like? Males have XY chromosomes, and it is a yellow-eyed male, which means that he will be carrying the lowercase b, crossing with a homozygous blue-eyed female. So she will have 2 capital B's. Now let's just move the page down. Next would be meiosis, and then gametes will be formed because we have gone through meiosis. If you don't understand this, go check out the videos on meiosis. So this is a monohybrid cross, so the gametes are nice and easy to get to. There we go. So now that we have the gametes, we can go into the process of fertilization. I have taught my learners to use the Punnett Square. If your teacher has taught you the other method, don't worry, we should get to the same conclusion. So let's get started with the Punnett Square. Now it really doesn't matter if you put the male or female on the side or on the top, you will get to the same conclusion. Let's quickly do this cross. Remember to write your dominant trait first. There we go. And then we will be able to get to F1. And then I always say genotype first, just because it's easier to copy it from that Punnett Square. Now, just before I carry on, remember to write meiosis and fertilization, because that will generally be a compulsory mark for a question like this. And then also, excuse me for that big blot there. So let me try a different color so you can see it. So then F1 and P1 also to be connected. You will get a compulsory mark for that as well. Let's just move the page down. Okay, so F1 genotype, what do we have? Let's start with this first block. We have a female that looks like that. Is she the only one? 1, 2 of those. And then we've got the first male. How many of those? 1, 2. A nice and easy one. So now you get to the phenotype. So what do we see when we look at this genotype? So we have possibility of 2 blue-eyed females, or a possibility of 2 blue-eyed males. If they ask what the ratio would be, the ratio would be 50% male, obviously, to 50% female, and both of those would be blue-eyed. So that's the first genetics question. It's really not that hard. I hope you got to the same conclusion. Now let's read that information again before we carry on to the next question. So after the larva stage, female mayflies usually live less than 5 minutes, while males can live a whopping 2 days. The average female mayfly lays anywhere from 400 to 3,000 eggs. You can often differentiate males and females by their eyes. Males will usually have much larger eyes than females, and males also have claspers beneath their tails that are used to hold the female while they fly and mate. Now let's look at that next question. They say, using the information provided, give 2 reasons why the mayfly is useful when studying genetic crosses. Now going back up there, I can already establish the 2 things that are used. So it's the fact that the females live less than 5 minutes and the males 2 days, and then the females lay anywhere from 400 to 3,000 eggs. So let me write that down and then I'll explain. So 400 to 3,000 eggs. And also the second one is a short lifespan. So looking at 400 to 3,000 eggs first, that means they have a large number of offspring that are produced. So the reason that is important is because the number of offspring is so large, you will have a very reliable sample size to get information on whatever you are testing with these genetic crosses. And then the fact that they have a short lifespan also helps with that, because that means they complete their life cycle within a maximum span of 2 days. So think about humans. If we were to do genetic testing on humans, so a male and a female that would have to produce offspring. So firstly, you would have to wait until they are able to have children. So that would mean at least a minimum of 13 to 15 years before they hit puberty. But we know humans generally only get children a bit later on average in life. So around about 20s, 30s, around there. So that would take very long to see the effects of the genotypes of the parents on then the genotype of the offspring and then the phenotype of the offspring, for example. So having a short lifespan helps a lot when doing genetic crosses or doing genetic studies to see the results of what you are studying. So that helps a lot. So the first one is having a large number of offspring, and then the second is them having a short lifespan. That's the end of this video. Hopefully you found it helpful. Remember to like and subscribe to stay up to date with any new videos that are posted.